Hello, hi, I'm Dr. Mahmoud Khalil. I will be giving you the topics of the molecular biology. So the topics include DNA and chromosome structure. So we'll describe and identify DNA and chromosome structure. The second topic is the DNA synthesis, in which we're going to explain the DNA organization and replication. And we'll describe the steps of DNA synthesis, replication, and organization of a eukaryotic DNA and the difference between eukaryotic and prokaryotic DNA. The third topic is about transcription and RNA processing through the process of RNA synthesis. We'll describe the structure of the RNA, the transcription of eukaryotic genes, and differentiate between eukaryotic and prokaryotic RNA. And as well, we will differentiate between eukaryotic and prokaryotic transcription. The next topic will be protein synthesis, and we will describe the steps and the characteristics of the protein synthesis. And we will define and understand the genetic code and the components required for translational steps of translation and the codon recognition by transfer RNA. For the last topic in molecular biology, it will be post-translational modification. We will understand what is post-translational modifications and explain their role in protein function. As well, we will describe the post-translational modification of polypeptide chains, trimming, covalent attachment like phosphorylation, glycosylation, hydroxylation, protein folding, and protein degradation. The main reference for the topics of the molecular biology is the molecular cell biology. It is by Ladesh et al. And published in 2016. So let's start with the nucleic acid, either DNA or RNA. They are linear polymers of nucleotides, contain the information for determining the amino acid sequence, and so the structure and function of all proteins of a cell. Actually, they are cellular factories that select and align amino acid in the correct order as the polypeptide chain is being formed. So it have all the information needed to select and align amino acid for a protein. This nucleic acid can catalyze fundamental chemical reactions in cells and regulate the expression of genes. So nucleic acid function as the brains and the central nervous system of the cell, while protein carry most of the functions they specify. For the DNA, the information contained in the sequence of each nucleotide is required to build all the protein and RNA of organism. DNA is a very stable molecule under many conditions. And the error rate of copying DNA is only 1 in 2.5 times 10 to the power 8 nucleotide. Very low rate of, very low error rate. So the exact replication of DNA, that ensures the eternity and the continuity from generation to generation. So you have the RNA, the second nucleic acid molecule. RNA or ribonucleic acid, actually it conveys the information on the DNA by copying it as an RNA. And they have 3 different roles. Messenger RNA carry the instruction from the DNA through a process of transcription, which specifies the correct amino acid during translation. While transfer RNA interpret the information in messenger RNA by the help of a ribosomal, the third type, the ribosomal RNA. So basically this slide shows overview of the 4 molecular processes we will talk in these lectures. So you can see the boundaries of the nucleolus, nucleus, and the cell. Initially, the DNA inside the nucleus performs the first molecular process, which is transcription, in which a molecule called RNA polymerase binds and tries to transcribe genes of DNA by the help of ribonucleotides, RNTP. Soon after that, a messenger RNA is formed, which is still immature, which is the pre-messenger RNA. It needs another molecular process, which is the RNA processing, in which RNA is converted from pre-messenger RNA into functional messenger RNA. This functional messenger RNA soon transported into the cytoplasm, where it got bound with the ribosomes and start the process of translation. So messenger RNA translation is the third molecular process. This ribosomes initially comes from the nucleolus, where the formation of the ribosomes is translocated into the cytoplasm and is waiting for the messenger RNA. So now we have three basic molecular processes: the transcription, the RNA processing, and the messenger RNA translation. The fourth process, actually, the replication. So DNA need to be replicated in order to be distributed on the two divided cells. In this process, it uses deoxyribonucleotides. Looking closely into the structure of nucleic acid, basically both of them are similar, but have some slight differences. So DNA, both of them are built of nucleotides, either adenine, thymine, cytosine, guanine, or uracil. All nucleotides have common structure. The AGC found in DNA and RNA, while T is only found in DNA and uracil is found only in RNA. So nucleotides actually are nucleosides that have either 1 or 2 or 3 phosphate groups linked to a 5' or hydroxyl group. And you can see from the diagram how the nucleotide is formed. It is formed of a base, which this base is either adenine, guanine, A, G, C, T, or U. This base is bound to a ribose sugar, pentose sugar. And this pentose sugar or the ribose sugar is linked to a phosphate group through a phosphoester bond. The structure of all this is the nucleotide of adenine. Both adenine with the sugar is called nucleoside, and the whole structure is called nucleotide. The ribose, it differs in the sugar. The ribose, if it's deoxygenated on the 2' carbon, in this case, this sugar will contribute in the forming of a DNA. While if it's normally a ribose, this will contribute in the forming of RNA. So this is one of the major differences between RNA and DNA, that the ribose in DNA is deoxygenated at the 2' carbon. Looking into the structure of the bases, you can see that there are majorly...
Doctor?
Sorry, yes?
Can you repeat the difference between the DNA and RNA? The one you said
The major
now.
difference in the chemical structure of the sugar. So you can see the difference here is highlighted. So at the 2' end on the carbon ring, you have here hydroxyl group, while here there is deoxyribose at the 2' end. This is the major difference. As well, of course, that RNA is single-stranded and the DNA is double-stranded. Looking into the structure of the nucleotide, generally they are classified into purines and pyrimidines. The purines are 2 rings that consist either adenine or guanine. Each of them consists of 2 rings, while the pyrimidines is only 1 ring. And the example is uracil, thymine, and cytosine. And this is the chemical structure of the nucleotides. Generally, this is the terminology of the nucleotides. And you can see...
Doctor, I have a question.
Okay.
Do purines bind to pyrimidines? Because as we know before, cytosine bind to guanine and adenosine bind to thymine. So
we
is
will
there
talk
a
about
link?
the binding. We will talk about the binding later on in the next few slides.
Okay.
So this table shows the terminology and how the nucleotide, if it's having only one phosphate, is called adenosine monophosphate, diphosphate, triphosphate. And this is a nomenclature for all the nucleotides. It's only for knowledge. So here, looking into the chemical structure of DNA and RNA, you can see that generally the nucleic acid have a sugar phosphate backbone. It is alternating sugar phosphate, as you can see, sugar phosphate backbone. Each of the sugar phosphate backbone, you can see that the base, which is either adenine or guanine or cytosine or uracil, purine or pyrimidine, 2-ring or 1-ring, is bound to the sugar. Looking closely, you can see these where is the sugars are, and the sugars is attached to each. The sugars is attached through a phosphodiester bond, and you can see the bases are here, C, A, G. In this slide, we're trying to show how the structure is written for the nucleic acids. You can see that there is always a 5' end on this carbon and the 3' end on the other end of the nucleic acid. Since nucleic acids are very long, so scientists agreed to do some simpler diagrams like the stick diagram. So you'll always have an end which ends with a phosphate at the 5' end, and then the other end will end with a hydroxyl group at the 3' end. And you can see the bases are above. Looking more simpler representation, you can see normally in references and over the internet, you can see researchers try to write the DNA in this simplest form. They only write the bases. They don't go deep for the structure since it is repeated. But make sure in any time you write or read a DNA sequence or RNA sequence, it should read from the direction of 5' to 3'. So again, it should be read in the direction of 5', 3'.
Doctor?
So, yes?
Please, can you repeat the stick diagram as a
The
field?
what?
Please?
The stick diagram?
Yes, please.
The stick diagram is only a representation in which the chemical structure, all these chemical structure are omitted and just represented by lines for simplicity. Soon after that, scientists adopt this way of writing the nucleic acid. They just write the bases starting from 5' to 3'. Looking into the structure of DNA, you can see that it is double-stranded. And in 1953, Watson and Crick proposed that DNA has a double helical structure based on the analysis of X-ray diffraction pattern of DNA, which was generated by another two scientists, Rosalind Franklin and Wilkins. So they found that the structure of DNA is double helix. And going to Shargaf's studies, they revealed that the percentage of adenine bases is equals to the percentage of thymine of T, while the percentage of G equals to the percentage of C. So that's mean A equals T in the DNA and G equals C in number in DNA. So the most accurate structure of DNA came from the high resolution X-ray diffraction studies, in which they studied the crystals of DNA. In this technique, they shoot X-rays on the DNA, and on a screen, they got the pattern of diffraction and distraction of these particles. This pattern will show the structure of the DNA. Looking closely into the structure, you can see that although they are double-stranded, but you can see that they are anti-parallel. So one strand will start from 5' to 3', as you can see from the arrow, while the other strand is in the reverse. And scientists, while understanding the structure of DNA, they found that in order to be stable, the structure had to be double-stranded and in anti-parallel strands. And they found that the DNA is a very stable molecule. Why it's a very stable molecule? Because there is a lot of hydrogen bonds. So you can see here that G binds to C by 3 hydrogen bonds. Can you see the arrow moving?
Yes, yes.
Okay. So you can see there is G binds to C by 3 hydrogen bonds, while A binds to T by 2 hydrogen bonds. So if you count the hydrogen bonds all over the DNA molecule, you'll find that there are thousands of hydrogen bonds, and that makes the DNA very stable. As well, there are 2 types of interaction, which is the hydrophobic interaction and van der Waals interactions between these stacked adjacent bases. So, I mean, in this region, in this empty region, there is a kind of interaction which is called hydrophobic and van der Waals interaction that keeps equal distance between the base bearing. And this gives another layer of stability for the DNA molecule. So Watson and Crick proposed this base bearing in which a larger purine binds with a smaller pyrimidine, in which A binds with T and G binds with C.
Doctor,
Yes?
where are the hydrophobic bonds?
The hydrophobic bond is not represented in this figure. Okay? It's not like it's non-covalent bond. Okay?
Okay, thank you.
Looking more deeply into the structure of a DNA, you can see that you need to know that most DNA in our cells are in the form of a right-handed helix. And we refer it as a B-form DNA, which is the normal form in every single cell in our body. You can see that the strands are anti-parallel. These strands are anti-parallel, and it is helix. Always you can see, and how you can notice that it is anti-parallel, because you have 5' end and the 3' end. Okay. Then you can see between these 2 strands, you can see how the bases, the steps of the base bearing. This orientation and this twisting of the DNA give rise to 2 grooves in the DNA. A major groove, can you see the major groove, the red one, and the yellow and the minor groove. So what is the function of this major groove and minor groove? The atoms on the edge of these major grooves, as shown by the arrow, are accessible from outside. So to make it clear, some proteins need to bind to the DNA. They can't bind to the strands. They bind deeply inside the DNA. So they need to move inside the DNA through the major and the minor groove. And of course, this major and minor groove, they are potential hydrogen donor and acceptor. That means they have the capacity to do a lot of bonding with a protein coming to do functions concerning with DNA. So this DNA binding protein can read the sequence of bases in duplex DNA by contacting atom through the major groove or the minor groove. These are the windows of interaction between any DNA binding protein and the DNA. So do we have only B-form DNA? Okay.
What does it mean in a right-handed helix?
Right-handed helix, if you twist something to the right.
Okay.
So the normal form, as we said, it is the B form. It is right-handed helix, right-handed helix. the complete turn in the helix, starting from here to here, it measures around 3.4 nanometer. And the distance between each two base pair around 0.34 nanometer. So you have around 10.5 base pairs per turn. That's the B form. While sometimes a form is formed. So what's the difference between the A form and the B form? A form arises in very low humidity or moisture. Once there is a decrease in humidity, the B is transformed to the A form. So what is the difference? The major difference, there is a large tilt of the pairs. So you can see here the base pair, these base pair, there is a tilt in the base pair, tilted. And instead of being around 10.5 pairs per turn, it will be 11 pairs per turn. So where this occur, this A form, sometimes in the cell, normally RNA found bound with a DNA, like in the process of transcription. RNA is bound to DNA while it is transcribed. Sometimes RNA binds with RNA in the cytoplasm or auto-bind to itself. So once this kind of interaction occur, it will be in the A form in cells or can occur in vitro. So that's a brief summary about the difference between a B and A form. So the overall proportion is different. Obviously, you have a short and broad A form, while B form is longer and thinner. Right-handed B form, both of them are right-handed. The base pair around 10, 11 in A. The major groove is wide with intermediate depths, while in A form, it is extremely narrow and very deep, as well as the minor groove differs between B and A.
Doctor, please repeat the previous slide because you went too fast.
Which one?
The previous one.
This one?
Yeah.
It's only showing the difference between B and A, showing that the difference occurred due to the low amount of water that's lead to A form, which is shorter and 11 base per turn. The A form exists in a structure like the RNA binds with the DNA, and as well RNA bind with RNA. Can you please just write down your questions on a paper, just mark the slide, and we can, I'll stop and ask if someone needs any question, okay? To just speed up because of the technical issues in this lecture at the beginning, okay?
Okay.
Okay. So you have this comparison. It's a good comparison between both of the B and A form. So another important characteristic of a DNA is the ability to bend. So DNA is flexible about its long axis. And this characteristic behavior allows DNA to bend in complex when bound with a DNA binding protein. So when a protein comes and binds with the DNA, it tended to bend the DNA. So why is bending is very important for the DNA? Because bending is critical, as we will talk later, about in the process of packaging of DNA in the chromatin. So talking for some of the important behavior of the DNA can undergo reversible strand separation. So what means reversible strand separation? That means the DNA can separate and then rejoins again under specific conditions. And this occurs during replication, of course, transcription of DNA. So the strands of the double helix must separate to allow the bases to pair with the bases of the nucleotide being polymerized to form new complementary polynucleotide chains. The process of unwinding or separation of a DNA strand is called denaturation or melting. And this process can be induced experimentally by increasing the temperature of a solution of a DNA. So as you can see from the figure, once we increase the DNA, increase the temperature of the solution, this will break the hydrogen bonds, and then there is a repulsion, electrostatic repulsion between the two strands. Why there is a strong repulsion between the two strands? Because the charge on the strands of DNA have a total of huge negative charge due to phosphate on these strands. So once the hydrogen bonds are broken due to the we increase the temperature in a solution, both of the strands now are single and they are covered by negative charge. So they start to repel each other. So the increasing temperature on a solution of DNA will separate the DNA strand. This will driven apart by electrostatic repulsion of a negative charge backbone of each strand. So you need to keep in mind now that both strands of DNA have a total charge of a negative charge, because this is important in the next few slides. So what is the melting temperature, Tm? Tm is the melting temperature at which DNA strands will separate. So it depends on the proportion of GC content in the DNA. Of course, if the DNA molecule have a lot of GC, that will increase the temperature needing to melt the DNA. And why is that? Because GC are bound to each other by triple hydrogen bonds. So once a molecule have a lot of GC pairs, it will have more triple hydrogen bonds. So you need more temperature to separate the DNA molecules. This first factor for Tm, or melting temperature. The second factor is the ion concentration. Of course, we are talking now, we are talking how to melt the DNA inside the lab, inside a test tube, inside an Eppendorf, inside the lab, in vitro. So we increase the temperature or decrease according to this factor to melt the DNA. The second factor is the ion concentration of a solution. When the positive ion concentration in the solution decrease, you will need a lower Tm to melt the DNA. Why is that? When positive ion is low, shielding is decreased. So this will increase the repulsive force between the strand. Of course, you know, as I told you in the last slide, that both strands have a negative charge. So when you have the positive ions in the solution, these positive ions will not be enough to neutralize the negative charge. So when they are very low in the solution, still both strands have a total charge, a huge charge of negative charge, and this will increase the repulsion. The third factor is you can use some agents that break the hydrogen bonds, like formamide or urea. So if you use this agent in a solution in the lab, you will decrease the melting temperature. The fourth factor is the extreme of pH. So you can use extreme pH. You mean this, you know the scale of the pH, it starts from 1 to 14. So we are talking about extreme of pH around the region of 1, 2, and around the region of 13, 14. So in this extreme of pH, either the extreme high pH or extreme low pH will decrease the amount of, the temperature needed for melting the DNA. At low pH, at low extreme pH, bases will be protonated. So it will have a lot of positive charge. So rebelling occur again. While at higher pH, the bases of the sugar backbone or the strand will be heavily negatively charged more than before, and again rebelling each other. So why we are talking about these factors, these four factors anyway? So assume you are in the lab now, and you need to melt the DNA. And we will discuss after that why we need to melt the DNA. And then you need to melt the DNA. And you found that to melt the DNA, you need a very high temperature, which is practically impossible, like 120 degrees. So you need to put agents like formamide and urea to decrease the TM needed for melting the DNA. So if you look here, we can monitor the separation of the DNA molecule by light at by using UV light at the range 260 nanometer. The bases of DNA in the state of duplex DNA, they absorb less UV, while in the state of a single DNA, they absorb high UV. So you can see from the figure, with increasing temperature, the double-stranded DNA is shown to be transformed into single-stranded DNA. This means that temperature is an important factor for denaturing or melting the DNA. While the single strand, since it's already single strand, not a lot of hydrogen bonding, so there is no major change in the reading of the absorbance. So it's a kind of a way to monitor the process of melting of the DNA. In order to know how much temperature is needed to melt the DNA, generally there is a correlation between the percentage of GC pairs in the DNA molecules and the temperature. And it is directly proportional. As long as the percentage of GC pairs increase, you need a higher temperature to melt the DNA. As an example, if we have a DNA molecule of this sequence, and you need to calculate the percentage of GC in this molecule, you can see that there is 5 GCs in a total molecules. If you count these nucleotides, you'll find that is total 10. So 5 GCs over 10 will give you 50% GC percentage. Looking back into the figure, you will see that 50% will give rise to 91 degrees Celsius. So for this molecule, you need a temperature of 91 degrees Celsius to melt the DNA. So to do the reversible process, you need to renature. Actually, you need to reverse what you have did during denaturation. You either lower the temperature, increase ion concentration, and especially positive ion concentration, and do neutralization for pH. If you used the extremes of pH. This causes the complementary strand to reassociate into double helix. Keep in mind that the DNA strand, which are not related in sequence, if you have different DNA sequences from different animals, they will not bind again to each other, and they will remain as a random coils and will never renature. So why we are talking about denaturation and renaturation of DNA? Because it can be used to study if 2 samples are related. And it can be used as well to isolate specific DNA molecules in a mixture containing numerous DNA sequences. So what is torsional stress in DNA? Torsion stress occurs in many bacteria and viral DNA, and as well in circular DNA of mitochondria and chloroplasts of the plants. And this torsional stress occurs during DNA replication and transcription of circular or linear DNA in eukaryotes. So what is torsional stress? Actually, DNA twists back on itself like a twisted rubber band, forming supercoils and induced what is called this torsional stress into the remaining portion of a molecule. Because of course, the ends of the DNA strand are not free to rotate. So all cells will have an enzyme responsible to relieve this torsional stress. There are two types of enzymes, either topoisomerase 1 and topoisomerase 2. Topoisomerase 1 bounds to locations on the DNA at random sites. These locations which have torsional stress, that means the DNA is coiled around itself. And this topoisomerase 1 will cut in one strand called the neck to relieve these torsional stress. And then after relieving the torsional stress, the two ends of the broken strand the same enzyme will rejoin the two ends. While topoisomerase 2, instead of cutting one single strand, it will cut a double strand. And once the torsional stress is relieved, the topoisomerase 2, the enzyme religates them. So in this figure, you can see how the DNA, this actually is circular DNA, how it looks when it's supercoiled. And of course, these are a major problem in the process of a replication of DNA or transcription, because DNA need to be linear or relaxed in order to be able to open up for replication process. So you can see this is a major problem, that it is need to be relieved by either the topoisomerase 1 or topoisomerase 2. Once it is relieved, you can see the neck here in the diagram, and the enzyme is relieved. It will soon transform it from the supercoiled form into a relaxed circle. And you can see here the enzyme, the topoisomerase 1, how it sits on the DNA and the neck, the single strand, pass the other strand and try to relieve the torsion stress. So again, what is torsion stress? It is a behavior that occurs to the DNA that need to be relieved in order for replication and transcription to occur. So let's talk about the organization of a DNA into chromatin. Beginning with the 21st century, molecular biologists came to the sequence of the whole genome of 100 of viruses, bacteria, and the budding yeast, SVC. By now, the genomic sequence is known for the fission yeast, S. bombay, the simplest plant, simple plant, rice, multiple multicellular, and a lot of other organisms, including human by now. They found that the DNA sequencing revealed that large portion of the genome do not encode messenger RNA. What's meaning they not encode messenger RNA? That means they can't form protein after translation of messenger RNA. So this region, or this non-coding DNA, is about 98.5% of the human DNA. So it's only about 1.5% of the human DNA only encode into a protein and functional RNAs. So what about the 98.5%? They collectively call it junk DNA. So these non-coding regions of the DNA are divided into three major regions, either introns, repetitive DNA, or some regulatory sequences in the DNA. And this repetitive DNA, either telomere or VNTR, a variable number of tandem repeats. That means it's a tandem repeats. So it's a kind of small sequences of DNA that is repeated all over the DNA. And the transposon DNA, which is like a small pieces of DNA that can jump in the DNA sequences. And this is a classification of the non-coding DNA, which represent around 98.5% of the human DNA. So the length of the where is the problem? Why we need to organize the DNA into the chromatin? Because the length of the DNA is a significant problem. It's around 2 meters in length and must be contained within nuclei which are around 10 micrometers. So there is some specialized eukaryotic proteins called histones that bind to the nuclear DNA and organize the DNA to fit inside the nucleus. In eukaryotes, the DNA plus these histones called the chromatin. And this can be visualized as the chromosome during mitosis. So what is the function of the chromatin actually? Obviously, the first function, it is helping in packaging DNA into smaller volume to fit in the cell. It helps in packaging the 2 meter long of the DNA inside a 10 micrometer nuclei. It maintains integrity of DNA during cell division. It helps the DNA in order not to be broken. It provides access, the chromatin as well, provide access organized in such a way to facilitate the transcription, replication, repair, recombination of DNA molecules. So these are the 3 major functions of chromatin. So how these occur? Here you have the single strand double helix DNA molecule. The first level in which the DNA is wrapped around histones. And this structure now is called nucleosome. This structure as a whole is called beads on a string structure. And this represents the first level of organization. Soon after that, these beads of a string undergo a second level of compaction, which is called the 30 nanometer fiber, in which these nucleosomes are wrapped again around itself to form a thicker structure that measures around 30 nanometer in distance, in length. So that's a thicker structure. Soon after that, this 30 nanometer fiber starts to make loops. So to make it clear, so the long 2 meter DNA single-stranded is getting shorter and shorter and shorter and thicker, shorter. Start from beads on the string, then 30 nanometer fiber in which the nucleosome are twisted around itself to form this fiber. And then this 30 nanometer fiber is twisted around itself like loops to form a thicker structure. Soon after that, there is a higher order of chromatin folding, leading to the interphase chromosome inside the nucleus. And these are the level of organization of the DNA to form the interphase chromosome, which is much shorter and thicker. Looking into histone protein, you have 5 types of histones. You have H1, H2A, H2B, H3, and H4. They are all rich in positive charge amino acid. And it is important to be positive charged because, as I said before, the phosphate group in the DNA are negatively charged. So this needs for the binding of the histone to the DNA. So you can see the structure of histone, that the DNA is wrapped around the histone. And each to form the nucleosome, as we saw before in the beads on the string. So this nucleosome, each nucleosome is connected to the other nucleosome through a linker DNA. And this linker DNA, it is naked DNA, which is not wrapped around nucleosomes, and it's around from 10 to 90 base pairs. This core histones actually is octamer. It's a 2 copies each of histone 2A, 2B, histone 3, and histone 4. The wrapped DNA is around 147 base pair of DNA. So again, the nucleosome is consisting of a core histones. Octamer consists of 2 copies of each of H2A, H2B, H3, H4. The sequence, the number of nucleotides that wrap the core histone is around 147 nucleotides or base pair. This will constitute the structure of the nucleosome. Each nucleosome is connected to the other nucleosome through a linker protein, a linker DNA, which is about to 10 to 90 base pair. So DNA of nucleosome will be stable and less susceptible to nucleases, which is enzymes that degrade the DNA, than in linker DNA. The last type is the histone 1, the yellow structure here. And you can see that histone 1, which is the linker histone 1, the fifth major histone, it binds the DNA at the point of the entry and the exit of the DNA wrapped around the histone core. And this completes the nucleosome. And this histone 1 is very important for stabilizing the chromatin configuration in 30 nanometer fiber. So here in this slide, we showed the structure of the nucleosome. Again, it consists of octamer of histones wrapped around the DNA of 147 base pair, connected nucleosomes through a linker DNA 10 to 90 base pairs, and the nucleosome is governed by histone 1 that closes down and complete the nucleosomal structure. These histones sometimes have variation. So you can see that histone 3 of sea urchin and calf thymus differ by only one amino acid, while that of the P and calf thymus differ by only 4 amino acids. So you can see that major difference between histone through the animal kingdom, there is not a lot major difference. And of course, you know, histones are proteins. The similarity in sequence among histones from all eukaryotes indicate that they are very similar in their 3D conformations. So there is only minor histone variant exist. Example, like the histone 2AX. So when this histone 2AX occur, normally if regions of a DNA got damaged, they need a repair process to occur. In this case, the histone H2A is replaced by a specialized other histone, which is called the H2X, which will help in the process of repairing the chromosome or DNA. While CNPAA, another type of histone that replace histone 3 and participate in the process of spindle microtubule formation during mitosis. While histone 3.3 replaces H3 in the region of the DNA where the DNA is actively transcribed. So these are 3 examples of variants of the histone that can occur in our body under certain circumstances. So what about the structure of 30 nanometer fiber? So again, we have beads on the string, the first level, then the second level is 30 nanometer fiber. 30 nanometer fiber, actually the structure is of 2 models. You have the first model, which is called the one-start helix or solenoid model, and this one is widely accepted. In this case, you can see that there are twisting helix rotation in which the nucleotide is arranged as a helix to form this structure of the 30 nanometer fiber. This model of the 30 nanometer fiber is called the one-start helix or solenoid model. And it shows the way of arrangement of the nucleotide. It's like a rope and you are twisting the rope. The nucleotide is after the nucleotide, N+1 nucleotide. And you can see here how this twisting occur. You can see this twisting occur. And the thickness now is 30 nanometer in length. While the second model is called zigzag ribbon or 2-start helix model, and it is strongly supported. So you can see a major difference is here. Although there is a helix, actually is a helix, but it's not like this one. Here the helix, you can think about it like a single strand is going around. But instead here, the nucleotide has to go in zigzag, and then this zigzag will go around to form a helix. So that's why it's called the 2-start helix model. Again, in this one, it's a kind of a rope, consists of a lot of nucleotides, and you do the zigzag. Each nucleotide is preceded by 1 nucleotide. But here, the nucleotide do a zigzag first, or arrange itself in a zigzag, and this structure all this structure will go into a helix. To make it clear, in this level, in a single round, you can see the two nucleotides because they are following each other. But in this here, you can see the following nucleotide because it is in another level, because there is a zigzag. So that's n plus 1 doesn't appear in the level, then n plus 2, then n plus 3 doesn't appear in the level, then n plus 4. But if you go here, you'll find n nucleotide, n plus 1, and of course, this one will be n plus 2, n plus 3, n plus 4. And here you can see how the two start helix appear. Okay, so again, this slide shows the two model for the structure of 30 nanometer fiber. One of them is widely accepted, while the other one is strongly supported. So during interphase, chromatin is dispersed throughout much of the nucleus, while during cell division, there are the folding and the compaction of chromatin produce the visible metaphase chromosomes. So chromatin exists in two forms. This actually a true picture under the electron microscope. You can see the regions, these are the regions of DNA that appear as beads on a string. And if you can see the nucleus at this stage, that means that these regions are actively being transcribed into RNA or being replicated. So these represent an active nucleus if you saw the DNA in the structure of a beads on a string. But if you see the DNA in the structure of 30 nanometer fiber, you can see that this one looks a much thicker than the beads because it's thicker due to the twisting. So in this region, DNA is not being transcribed or replicated. So this showed the contrast between, or the correlation between the structure and function. So when the DNA in the form of a beads on a string, that means it is actively transcribed or replicated. While when it starts to be compacted, that means that the DNA is closed. That means there is no transcription or replication. So looking closely about the levels of compaction, the chromosome condense and decrease in length, generating the famous chromosome, what is obvious in the metaphase. Each chromosome actually contains one linear DNA molecule. For this process to reach, to happen in the chromosome, a specific protein called condensing protein, and actually it's composed of a lot of other protein, which is called the structure maintenance of chromosome, SMC proteins. All of these contribute to the process of chromosomal condensation. And of course, this process uses energy from ATP hydrolysis. So what is the event of mammalian chromosome condensation? Of course, we talked about the 30 nanometer fiber. And if you remember, we said that after the 30 nanometer fiber, there is higher order of condensation. This 30 nanometer fiber is followed by more condensation to form the chromonemma fibers, which are around 130 nanometer fiber. Then another level of condensation, which is called the middle prophase chromatid, which are around 250 nanometer in thickness. This is followed by the metaphase chromatid, which reach 750 nanometer. And this actually what we can see by our eyes under the light microscope. But all these stages cannot be seen under the light microscope and need an electron microscope to see it. Only we start to see under the light microscope the structure of the metaphase chromatid. Of course, after the chromatid is formed, disjoined to each other, and there is a central constriction which is called centromeres to form the typical chromosome. So you can see here this table only for knowledge. You don't need to memorize the numbers. You can see here the examples of different organisms, and the number of base pairs, and the number of protein they generate by translation, and the number of chromosomes. You can see bacteria like Escherichia coli have only one chromosome, while the fruit fly Drosophila have 6 chromosomes, while Homo sapiens, the human, have around 24 chromosomes. It's not strange to say that it's 24 chromosomes, because this table shows the total number of different chromosomes, not actually the number of chromosomes inside the cell. Because as we know from our previous studies, that each cell contains 23 pairs of chromosomes. Okay, the numbers here are the numbers of the total different chromosome. So that means in human, each cell normally contains 23 pairs of chromosome. For a total of 46, so you have 22 of these pairs called autosomes. So you have 22. Then looking into the sex chromosome, which is the 23rd pair, it's called the sex chromosome, which is different from male and female. You have an X and Y. So you have actually 22 autosomes, and the X and Y will be 24 chromosomes, okay? So here we reach the final shape of the chromosome with 2 chromatids connected together by a centromere, and this is the typical metaphase chromosome. So where chromosome exists inside the nucleus? By this experimental figure, you can see that they fix a type of cells called the fibroblast in human in the interface, and they labeled the chromosomes fluorescently to emit fluorescence in situ, that's mean in its location. Specific, they labeled only chromosome 7 and chromosome 8. And you can see that each chromosome stayed, because we have a homologous chromosome, a pair, so you can see the location of chromosome 7 and chromosome 8 inside the nucleus. The rest of the nucleus is stained by blue. So in this experiment, they fluorescently labeled this chromosome to identify its location. In a diploid cell, so each of these 2 7s or 2 8s is restricted to territory within the nucleus. They are not stretching around through the entire nucleus. Making more sophisticated, scientists will able to stain every chromosome in the nucleus. And you can see here, every chromosome is stained by the same method, fluorescently labeled, and they managed to identify where every chromosome is localized in the interphase cell, interphase fibroblast. And you can notice that each chromosome occupies its own territory inside the nucleus. Of course, you don't need to memorize the numbers of the figure, but you need to understand the principle of the process, the principle that each chromosome is not stretching throughout the entire nucleus, but it's only localized to specific territory inside the nucleus. So going deeper for how they know the abnormality in chromosomes, so certain dyes can selectively stain some region of the metaphase chromosome. They can stain it more intensely than others. And this produce characteristic banding pattern that are specific for individual chromosome. So this banding technique can help if the chromosome is of the correct shape, correct size, similar shape, similar size. And in this technique, they use a kind of fluorescent dye that they attach on the chromosome to get fluorescence, and they can be easily seen. And this, when this sample is observed under a fluorescent microscope, the information is conveyed to a computer where a special program gives a false color to each type of chromosome. And then these computer graphics allow the 2 homologous of each chromosome to be placed each other, and they can number it according to their decreasing size. So basically, we are describing the process of karyotyping. They label the different chromosomes with a fluorescent dye. that they detect by a fluorescence microscope, and then we do some graphics and putting color for the chromosome for identifying the number of chromosome and similarities and size in order to develop a karyotype for each individual. This chromosome painting is a powerful method for detecting chromosomal abnormalities and as well for comparing karyotype of different species. So you can see here how chromosome can be stained or fluorescently labeled with different fluorescent dye, and then they can be aligned to understand the, to show the homologous pairing. In this alignment, you can see that if there is some chromosomes are abnormal when comparing each homologous chromosome, you can see that this karyotype is of a male, because you can see here X and Y chromosome. So why this technique is very important? You can see here that you can develop this technique to observe abnormalities in chromosome. Normally, this is chromosome 9, and this is chromosome 22. Okay? So this normal chromosome. If something occur like translocation of a part of chromosome 9 to chromosome 22, will generate a chromosome that normally called a derivative 9 or der9, and another one which have a part of chromosome 9 which is called derivative 22, and normally it is called Philadelphia chromosome. So this characteristic translocation between 9 and 22 is occur in chronic myelogenic leukemia, and this Philadelphia chromosome actually it's shortened chromosomes and abnormal, and will have, so this individual will have two abnormal chromosomes, a shortened chromosome 22 and the abnormal long chromosome 9. This, as I said, result from translocation between normal chromosome 9 and 22. And this can be used by the fluorescent label to identify the difference. So you can see here by fluorescent label, they identified specific to chromosome, for chromosome 9, they identified the normal chromosome 9. And as well, they identified the normal chromosome 22. This one have a green fluorescence, this one have a orange fluorescence. And they as well identified the translocation, because here you can see a long chromosome abnormally have a green signal. That means there is a region have been moved from the normal 22 to the 9. And as well, you can see how these have a mix between orange and the green. And this represents the Philadelphia chromosome. So here, we're trying to elaborate on the use of the fluorescence labeling to understand if there is abnormalities in the disease, in the chromosomes. And you can pinpoint where the translocation occur. The obvious example here, the translocation between chromosome 9 and 22. And this concludes our lecture. You will find what I have told you in this lecture in chapter 258, and these are the pages from the reference